Alex has some questions that ask you to identify phospholipids, fats, sphingolipids, and functional groups within these molecules. And it's not that the names of these molecules are so terribly important that you know, but I'm hoping you will see how learning functional groups has helped prepare you to understand more biochemistry, because these are all important molecules in our cells. So there are some facts about them. These phosphoacylglycerol molecules, or phospholipids, they all have phosphate in them. So we're going to look for groups that have P connected to O's, and they're going to have negative charges on them. And I'm not drawing everything about them because they connect onto other parts of the molecules. They get divided into groups called cephalins and lecithins, and you can recognize those because cephalins have a terminal ammonium group. So we're going to look for an NH3 with a positive charge attached onto one end. Lecithins have trimethylammonium groups. So instead of just this ammonium connected onto the rest of the molecule, we're going to have 3 methyl groups connected. So these things you know about functional groups will help you identify and understand what's meant by the terminology. When someone says it's a cephalin and you're like, what's a cephalin? And you look it up and it says it has an ammonium group on the end of it, you're like, oh, I know what that is. They'll have a glycerol molecule that's going to be hidden in ester bonds, and one will go to the phosphate, and the other two are going to go to these ester bonds with acyl chains. And they'll have 2 acyl groups, long hydrocarbon chains. Triacylglycerols, what we typically call fat in animals, or they make oils in plants, have glycerol. 3 carbons, 3 alcohol groups. But instead of alcohol groups, they have esters, 2 long chain, well, not necessarily long, but 2 fatty acids with a lot of carbon and hydrogen atoms. And then there's one more group, sphingomyelin, and I'm not going to try to memorize the structure. It looks a little bit the same, but instead of glycerol, it's got these 3 carbons you can see how it's a little bit like the glycerol with the oxygen, which then goes to a phosphate group, and an alcohol coming off on the other end, but then instead of an alcohol it has an N, which goes into an amide and a long hydrocarbon chain. And this alcohol, instead of becoming an ester, this is directly connected to a long hydrocarbon chain. So looks a little bit similar, but there's some significant chemical differences in the connectivity here. The carbon-carbon bonds are much stronger than carbon-oxygen bonds. Not as many things can make them or break them. So sphingomyelin has similarities to acyl chains, a phosphate group, just like phosphoacylglycerol, amide, which is different but similar to an ester. But by knowing your functional groups, you can distinguish among all of these. So let's take a look at a specific Alex problem. Okay, here is our first molecule. We can start looking for things. Instead of ester linkages like the others have, it has 1 amide linkage, a phosphate group, that's an alcohol, and then at this end we do have a trimethyl ammonium group. So let's look down, this is our giveaway here. The amide is telling us it is sphingomyelin. So we go down and make our choice. Sphingomyelin, check it, and we can move on to the next part of the problem. Highlight the phosphate in this structure. So you might be asked to highlight different groups. And I will highlight the phosphate, but I'm going to do it last. If I were highlighting a hydroxyl group, I might have to highlight this whole thing. Let's reset it. The alkene, I'd highlight the bonds and the carbons in it. If I were highlighting the amide, I'd start at the nitrogen and highlight everything up to the oxygen. That would be what I would highlight for the amide. The phosphate, I would highlight all the O's and the bonds and the P. The trimethyl ammonium, I would highlight the nitrogen, the bonds, and the carbons, the methyl groups. So I'll finish with the one I'm supposed to. Got all the O's, all the bonds, and the P, and I can check it. And we're good to go to part 3. Highlight the amides. It's a little different from highlighting an ester. For it to be an ester, I would need to include the carbon here beyond the what would be an O, but it becomes an amide just by attaching an N to the carbonyl, and I don't need to go beyond that. So check part 3. And any of the other problems where you're shown a phosphoacylglycerol or a triacylglycerol will have similar parts where afterwards you have to identify the functional groups within that molecule.